want to thank the organizers for inviting me to show you the data that we have. And I also, is it not loud enough? Actually, I would rather not use a microphone. And I'd also want to say it is great to see all the families here, because really the reason we're doing this work is because of all of you, and you motivate us and keep us going. And thank you for the samples that we're going to be taking from this meeting. So the work in the lab is really focused on identifying efficacious therapeutics that can be used to treat CTNNB1 syndrome. And obviously you all know this is caused by sporadic mutations that occur. They're pathogenic, they're loss of function. There are different types of mutations that span the entire gene. So we're talking about multiple different mutations giving rise to this syndrome. And basically, the core problem is, so you have two genes, CTNNB1. The genes encode the protein beta-catenin. So one of the gene alleles now is not really producing a functional protein. So you basically have like 50% of this protein. So that is what we call your core molecular pathology. And beta-catenin, you've been hearing a little bit about it. It's expressed in every cell of the body at every stage of life. And it is a really important protein for proper maturation and function. And I'm going to tell you about its function predominantly in the brain and in nerve muscle synapses. So that is the core pathology that we're targeting on fixing. And beta-catenin, or CTNNB1, is a well-known, what we call a high-confidence risk gene. If you look in the Simon's database for human disorders, it is one of the high-confidence risk genes for intellectual disability and autism. So it's commonly known that this is a really important gene in the body. And you heard a little bit from Kellen's talk. There are several other human gene mutations that cause intellectual disability and autism that actually converge on that beta-catenin Wnt signaling pathway. So it's really playing an important role. It has really upstream players and downstream. It's essential. If both copies of that CTNNB1 gene were mutated, it's not compatible with life. There would be no fetus born. So basically, treatment's lacking. We all know that. That's why we're here. We're trying to fix this. And it started really because we know very little about what are the pathophysiological changes that happen when you don't have enough beta-catenin. And really, to understand it, we didn't have preclinical models. So I started working on this after families reached out to me in late 2019. And so we decided to make preclinical models so we could start to look at it. So the first thing we did was to make a mouse model, and it's called a germline heterozygous mouse model, where we knocked out one of the alleles of CTNNB1 in the entire body. So it's a full body heterozygote. We've also been working with different human cell models. We started with a healthy human cell line that we took out one of the CTNNB1 genes. But most important, we're now working with patient cell models, and that's going to be the last part of the talk, and I think that's the most exciting, most relevant, and we've got some really great data I want to share with you. So from the work we're doing, we're learning new things about what happens when you don't have enough beta-catenin. We're finding novel molecular and functional changes in parts of the brain. I'm not going to get into that kind of detail right now. I'm going to give you a more big picture. But we have efficacious therapeutic treatments that are working in both our mouse and our patient models. And I'm going to focus most of the talk on the small molecule. I had a slide, but I took it out because it's on the work with Jeff, but I'm going to describe it. I was thinking, should I upload it, but there's not enough time. So let me tell you a little bit about beta-catenin just to bring you up to date on thinking about it in the nervous system. People are talking about it in different parts of the body. It has two critical pathways that it functions in. This is a little drawing, it's a schematic of a synaptic connection. Synapses are those sites of rapid information transfer where a nerve contacts its target, sends information. Yes, throughout your brain, the synapses of your brain control everything you do. So the synapse here, this is representative of an excitatory synapse in the brain, but it would also be any synapse where the nerve is contacting skeletal muscle. And beta-catenin is going to function at that synapse. It interacts with these adhesion molecules. In the brain, it's called neuronal cadherin. That brings the presynaptic side in alignment with the postsynaptic side, and beta-catenin stabilizes that contact, but it also controls it. So it controls the number of synaptic connections you have, and when you undergo learning and memory, it enables the synapse to get bigger, to put more receptors in, to be more responsive to activity. So it's critical for synapse number, stability, and plasticity central to learning. That's up at the synapse near the membrane. Beta-catenin also functions, you heard, in the Wnt pathway. It travels into the nucleus. And in the nucleus, it controls expression of genes in response to Wnt signaling. Kellen mentioned Wnt. In the brain, Wnt, that little soluble molecule, is released with activity. So when your children are being stimulated, when they're learning, when they're doing exercise, Wnt is being released. It's actually transiently increasing the amount of beta-catenin in that cell, which is why I always say keep your children active, keep them stimulated, keep them going. Don't stress them, but you can actually increase beta-catenin levels with just physical mental activity. It may not come up to wild type, but it's beneficial. So it goes into the nucleus and it turns on expression of certain genes that are also really important, controlling cell proliferation. That's why beta-catenin, if it's excessive, can lead to cancer. But it controls cell fate, cell identity. It also controls axon guidance, the wiring of the brain, and it controls plasticity during learning and memory. The products of that Wnt pathway play those roles as well. So you can see how important those two functions are. So we said, we know a lot about beta-catenin Wnt pathway. My lab has been studying it for a very long time, gain of function, loss of function models in the mouse. And you heard about the Wnt pathway, and I'm going to point to glycogen synthase kinase. So this is an enzyme that also in every cell of the body, it's part of that Wnt pathway. And when you have that activity and the Wnt is going to be released, what happens is it inhibits that enzyme. So this is an enzyme that's always active. And what happens with Wnt signaling is it's transiently inhibited. So when it's active, I said it does this change to the beta-catenin protein. It phosphorylates it. That targets it to be degraded. It's a way that your body controls your levels to keep them in that healthy range. And with activity and learning, the levels go up a little bit. They can go to the synapse, increase the synapse size. It can go into the nucleus, turn on Wnt responsive genes. That's really critical. We know about this. It's an endogenous protein. We target it. How do we target? My collaborators at the Broad Institute, that's a part of Harvard and MIT, they've been working on this pathway for years, and they have this beautiful high specificity inhibitor molecules, a library of these small molecules that inhibit GSK3. And we got in touch with them. It was originally Florence Wagner, who was the director of their therapeutic development branch. We now work with Michelle Weaver, who is going to be here, but he's home with his newborn. And we are working with two of their drugs that they have developed that are these exquisitely high-sensitive inhibitors of glycogen synthase kinase. Small molecules that show brain exposure when you deliver it to the body systemically. Also, they're potent, and we're looking at the metabolic stability, although they're really important properties for a drug. So members of my lab, some are here, John, Crystal, on the paper, Nuran, who's a new member of the lab, did this work that we just published in 2024. This is using the small molecule inhibitor in the mouse model. So I remember I told you we had to generate preclinical models. So we made the full-bodied germline heterozygote, and the beautiful thing about it is it displays key features of the disorder in humans. We've learned new things about it from molecular functional, but most important, we've used it to test those small molecules, and I'm going to tell you data that it corrects the phenotypes. Oh, yeah, I'll come back to it. I always do this. I always forget. I want to put that on the top, which I always go right to the science. These are the groups who we're associated with. So we started this work because we were involved and contacted by CTNNB1 Connect and Cure. We're closely associated with the Broad, I told you, with the small molecule work. Based on the mouse model I just told you about, we have a great agreement or research collaboration with NIH NCATS. This is their translational development branch, where they work with us to improve the properties of the small molecules to get a lead drug candidate and do the high-level studies that we need. And the last group, we're fortunate to be Oxford Harrington scholars. We were chosen as rare disease scholars. These are people that are high-level advisors that have had like 30 years of experience. I have 2 advisors, each of them over 30 years of experience in major pharmaceutical companies. They look at our work, tell us what we need to be the essential preclinical work, and connect us to like big pharma and to venture capital. Okay, so we worked at the mouse. We showed, yes, it has 50% of the levels of beta-catenin protein and mRNA. That looks good. It shows all the key phenotypes that we see in the patients. We've looked now at different parts of the brain. We've looked at the spinal cord. We've looked at skeletal muscles, muscle in the leg, the diaphragm. I'm not going to get into the details of the molecular and functional changes, but we learned novel changes that are caused by insufficient beta-catenin. The mice show reduced spatial learning. They show reduced motor learning, reduced coordination, and muscle weakness. The most important thing is whether or not we can correct it. And so what I'm showing you here is a drug-treated mouse. This is the high-specificity GSK3 inhibitor that we got from the Broad. The blue is the wild-type littermate. You always compare that. The red is the heterozygote mouse. These are protein levels. And the gray is the drug-treated mouse. The gray bar is up to the same level as the wild-type. We have normalized the level of beta-catenin. It's not too high. It's not low anymore. It's normalized, which is really our target, so that's really important. It corrects the molecular and functional changes we found. It corrects downstream beta-catenin functions in the interactions with cadherin and in the Wnt pathway. I'm just not showing you all the data because of time. And it corrects the phenotypes. The learning, the gray bar, is at the same level as the blue bar in the tests for spatial learning, in the tests for grip strength, it's up, significantly improved, and in the rotarod, which is a test for balance. So we are able to rescue the phenotypes. And one of the things I think is really important to point out, relative to the question we had earlier, how early do you treat? We started the treatment in 6-week-old mice. We treated for 5 days, then tested them. 6-week-old mice are equivalent to late adolescent young adult in a human. So they were fully symptomatic before we even started treating them, and we still got this kind of level of recovery. And that's because the interactions at the synapse and the interactions in the nucleus, they're very plastic. They're important in every stage of your life. So you can have a window where you can improve the phenotypes even with late-stage treatment. So what's the summary of the mouse work? It's the great model. It shows key features we see in humans. We've identified an efficacious small molecule treatment. We treated symptomatic mice and got beautiful recovery. We normalized the beta-catenin protein levels. That's our target. We treated wild-type mice with the same drug, same treatment, and saw no adverse effects for length of treatment, no change in learning, no change in beta-catenin levels. So we're really optimistic about it. And this is the work that got NIH involved in wanting to work with us to turn these into a lead drug candidate to go to a clinical trial. So this is great. We're really excited. We're going to do more in-depth studies about safety. But will it work in a person? You know, really, that's our goal, right? A patient. And so I'm going to show you the work we're doing in patient-derived cells, because I think, like I said, that's the most relevant model. So we are working with 6 different stem cell lines. So when your children donated blood to, like, Simon's Searchlight, they take the white blood cells and they get them de-differentiated into what we call pluripotent stem cells. And then you can get those pluripotent stem cells and differentiate them to all different cell types of the body. So we got 6 different, what we call iPSC, pluripotent stem cell lines. They are, the ones that we have from Simon's are all nonsense and frame shift mutations, which are the predominant mutation types that we're seeing in the patients. And we found that in this proliferative stem cell, the beta-catenin protein levels are reduced about 50%. It looks a lot like the mouse. We take those stem cells and we differentiate them into excitatory neurons that you would find in the brain of a human, into the cortex. We still see 50% decreases in beta-catenin protein levels in patient-derived brain neurons. We looked at a downstream function in the Wnt pathway. We're starting to do that in more depth. It's also decreased. The beta-catenin functions are down. People worry, if you have the mutated allele, is it producing a protein? Is that going to have any negative effect? Is it going to affect the wild type, the healthy beta-catenin protein? We've now looked at these 6 patients and done a study to look for any product from the mutated allele, and it's not present. So these patients tell us that they have 50% protein, looks like the full length, and it's just like the mouse model as far as that. So that's pretty exciting for us. We're happy about it. Does the drug work? And the answer is looking really great. So we're now working with 2 different high specificity inhibitors, GSK3 inhibitors. The lighter gray is the one that we used in the mouse. The darker gray is another one that the Broad has developed that is more than 10-fold more potent. We're using it at more than 10-fold lower concentration. They are both working to increase beta-catenin protein levels in all 6 patient lines. We're starting to look now at beta-catenin function in that Wnt pathway. It's looking like that's also increased by the drugs. So we're getting positive effect in patient-derived neurons with the same drug treatment. And one of the newest things that we're doing that we're really excited about, I would say, so I try to remember all the things, we're starting to collaborate with a colleague who's taking the pluripotent stem cells that we have and differentiating them to spinal cord motor neurons. And we're also going to start to work with organoids, which are more than one cell type. Now it's excitatory, inhibitory, glial, cortical organoids and spinal organoids to make it more like a model with all the cell types and the interactions, and to treat them with the drug and show whether or not it's effective. But one of the things we're doing that we're really excited about, and this came out of the Oxford Harrington, they said, we really want you to show target engagement, and we want you a way to monitor the patient's response to the drug. So we're taking white blood cells that are isolated from patients. And first, we're monitoring what are the beta-catenin protein levels down, what are the beta-catenin protein levels in the red. You see that they're down compared to, this is from the mouse. This is wild-type glutamate. Yeah, this is the mouse one, not the patient one. I have to check my slides. The blue is the wild-type glutamate mice. Red is blood cells from our heterozygote mouse. Protein levels are 50% down in blood. We take the blood cell, put it in culture for a day, treat it with the drug for a day, look at beta-catenin levels, and they're up. And so this is evidence that we have target engagement and that we can follow beta-catenin protein levels in an individual who would be treated with the drug, which is really important, because if we have to change dose or we have to change frequency, we'll know it. It's a non-invasive assay. We can follow it. And so we are moving forward to test this in patient samples. And today, many of you and your children were great, and you got blood donated, and we are taking several of those samples back to the lab right after this meeting to do this in the patient-derived blood, because it's going to be a great way to say they're responsive to the drug and we can follow it. And so we have really important preclinical data that shows that we have a small molecule therapeutic that looks efficacious and we think it looks safe. I'm going to give you a 5-second overview of the work I'm doing with Jeff Collar. So Jeff and I started interacting a couple of years ago when Jeff, I know, had this new treatment for what he calls haploinsufficiency disorders. You have one functional gene, one not functional. And typically, I think a lot of you have heard about these antisense oligos. And so the goal here, Jeff has this novel approach where what he does is he said, here you have this one healthy copy of CTNNB1. Because I went to him, I said, I have this mouse, we can test it in my mouse, and now we're testing it in the patient cells. You have one functional copy. He has an antisense oligo that's designed to be a little guide that goes only to the CTNNB1 untranslated region at the 3' end. High specificity, doesn't see any other RNA. What he attaches to it is what's called a poly-A tail. All of your messenger RNAs in your body have poly-A tails, and they control the stability and the translation, the protein made from that RNA. Over time, the poly-A tail gets degraded because you don't want to keep making protein constantly. What Jeff does is he has his little guide that brings in another poly-A tail to the CTNNB1 mRNA, so it stabilizes the mRNA and it makes more protein than would be normally made from your healthy wild-type copy. And we tested it in different human cell lines, but we tested it in the patient-derived cells that we have as neurons, and it increases beta-catenin protein levels. So it's a different therapeutic, but it also works really well. We don't have excessive levels. Our goal right now, I think we've gotten as high as almost 1.8-fold. We want 2-fold. He changes the design a little bit, and we're working on that. And we're really excited because that's another therapeutic, and it's very specific because it's only targeting CTNNB1 mRNA. So we think that it's going to be very safe and well tolerated. So those are two different therapeutic approaches that we're really going after. And this is basically, again, the groups I work with, the funding, support from the families has been fantastic, and I thank you all.